Hello students, welcome to Lab 14. We're going to be talking about karyotyping. Basic introduction for karyotyping. This is just a visual representation of all the chromosomes within a cell, right? So you have all of them lined up together within a visual picture, right? So if we say that we see all of the human chromosomes together, we have 23 pairs, one set by the mother and then one set by the father, right? So then we have each chromosome has a number. Chromosomes 1 through 22 are referred to as autosomal chromosomes. Okay. All of these are autosomal, which means that they can duplicate themselves. Chromosome 23 is the sex chromosomes. That determines our sexes. So we have one X and then one Y for the male sex, then one X and then the second X for the female sex. We have chromosomal arrangement. In a karyotype, homologous pairs of chromosomes are arranged by size, what's called G-band patterns, and then their centromere location. So those G-band patterns are just there to indicate the genes, right? Where the genes are, and then where their alleles are located per gene. So homologous pairs of chromosomes, if you have the sister chromatids together, each of those sister chromatids are linked via their centromeres. So where that centromere location is located, that's also what they're arranged by. So in this karyotype, if we set all that karyotype up, sometimes we can see what's called chromosomal abnormalities. So we can detect various abnormalities, such as aneuploidy. This is any deviation from the normal chromosomal number. Some disorders involving cell having an extra chromosome, or 2n+1. These are called trisomic cells. So Down syndrome is a trisomy syndrome. Down syndrome is a disorder where you have an extra chromosome in the 23rd pair. I'm sorry, the 21st pair, sorry. Aneuploidy, or autosomal chromosome disorders, happen within the 21st to 22nd chromosomal pairs. So this is an autosomal chromo disorder. Almost all of these disorders are lethal. Almost all of them. But with modern medicine, these are livable. That being said, it's sterilized. It's at this point it's like I said, with modern medicine we can kind of get over this, but with a third chromosomal pair, this is a sterile individual, because as a third set, you can't break 3 in meiosis. You can't break 3 chromosomes in meiosis and make a full set of a haploid set. It's not possible that way. So this individual would be sterile. They wouldn't have the ability to make a child. All right, other types of aneuploidy involve the loss of chromosomes. So this is 2n minus 1. This disorder is called monosomic because of the individual that has only one copy of a certain chromosome. Alfie's syndrome is monosomic, occurs in the 9th set of the chromosome set. Okay. There are several non-lethal chromosome disorders. Usually these occur in the 23rd set. So Turner syndrome, this is where a female lacks one X chromosome. This is called XO. Or we have Klinefelter syndrome. This is a male with an extra X chromosome. Klinefelter syndrome usually occurs where a male can have female-like characteristics. Sometimes we have female with an extra X chromosome. This is called triple X. And then we have Jacob's syndrome. Jacob's syndrome is a male with an extra Y. Sometimes this one occurs where males get a little bit more testosterone. In meiosis, the process of non-disjunction may cause a gamete to have more or fewer than normal number of chromosomes. So this is where you get those extra chromosomes, or this is where you get those chromosomal abnormalities. So non-disjunction occurs when homologous chromosomes fail to separate during meiosis, or when sister chromatids fail to separate in meiosis 2. The timing of non-disjunction will affect the genetic makeup of the gametes. So during normal meiosis, we have all of the gametes going together and they'll split apart into normal haploid cells. So if non-disjunction happens in meiosis 1, more DNA will be pulled to one side during the first, or before the first division in meiosis 1, and then during the second division in meiosis 1, you'll have more DNA in half the gametes than the other half the gametes. So you'll have a lot of DNA in two gametes and a little DNA in 2 gametes. So all of the gametes are affected. If you have non-disjunction happening in meiosis 2, you'll have half the gametes that are non-affected, but half the gametes that are fully affected, where it splits the first time, and during the second split, more DNA is pulled to one side than the other side, leaving one gamete with more DNA than the other DNA, or than the other gamete. So it's better to have non-disjunction in meiosis 2 than in meiosis 1. but it's not always available. So let's try to see if we can analyze some karyotypes. So in this one, is this autosomal or sex chromosome disorder? Well, let's see where the irregularity is. Which one has more or less chromosomes in it? Should be number 18. 18 has more chromosomes in it. So 18 has 3 chromosomes in it. And if you look up what trisomy 18 is, that's called Edwards syndrome. What is the sex of the individual? That would be male. So you can use this to go over the lab assignment that I gave you. There should be about 4 or 5 of these same karyotype analyses. Okay? You're going to go over those and kind of answer these same type of questions. All right? Do that and turn those in. And if you have any questions, please let me know. All right, I will see you later.